Hello, my name is Danae Bartke, and I'm a classical HCU patient and the Executive Director of HCU Network America. This month's RareX Homocystinurias Data Collection Program chat is a little different. Rather than focusing on one of the domains in the program, we are focusing on how RareX can help drive the standard of care and understanding for the homocystinurias. Today, I'm joined with Kimberly Chapman and Suzanne Hollander. Suzanne Hollander is a metabolic dietitian at Boston Children's Hospital. She works with children and adults with various inborn errors of the metabolism in the clinical and research setting, helping individuals achieve health and nutrition goals they have for themselves and their families. Susan is also a member of the Genetic Metabolic Dietitians International, otherwise known as GMDI, where she has participated with the GMDI CERN Nutrition Guidelines Project. We are also joined here today by Dr. Kimberly Chapman. Dr. Kimberly Chapman is a metabolic geneticist at Children's National and an assistant professor of pediatrics in the integrated systems biology at George Washington University in Washington, D.C. in the United States. She specializes in taking care of all ages of individuals with defects of methylation, homocysteine, and propionate metabolism. Dr. Kimberly Chapman was also a member of the EHOD Guidelines Committee and was responsible for gathering the data from a review of the scientific literature that led to the guidelines for the diagnosis and management of cystathionine beta synthase deficiency that was published in the Journal of Metabolic Diseases. Thank you, ladies, for joining me today for this great conversation here. It's been really exciting kind of starting to dive in to the data collection program we have through RareX for the homocystinurias. So today we are going to be talking about how those how the data collection program can drive standards of care. For anyone who's listening in live through Zoom, feel free to drop your questions into the chat and we will address those in a timely manner. So my first question here is it's more of an educational piece. It's rather than a question, because there's so many different ways patients can participate in research. And so I thought we would start with trying to figure out how these things differ. So RareX, it's a data collection tool with surveys. And so would Kim or Susan, would one of you be able to talk to how this would differ from an actual clinical registry?
So do you want to do this, Susan, or I can?
You go for it.
Okay. So when you think about information that's important to understanding a rare disease, you can think of it as, how do I gather that information? And so we're going to talk about how do you gather information, and we'll talk a little bit about natural history studies, and we'll talk a little bit about clinical registries, and we'll talk a little bit about how things become gathered and understood. So historically, what happens is individuals with rare disease, they'd see their physician, they'd get diagnosed, and their physician would write a paper about it. Okay, so that's the historical thing. So when we talk about the original descriptions of these disorders, somebody wrote a paper. And then after you write a few papers about what you think of as the common phenotype, the common way somebody is, then the papers that end up in the literature are papers about rare complications, so things that aren't so common. But remember who's doing this. The physician is doing this, okay? So the physician is looking and figuring this out. The patient has some insight that the physician's not going to have. It's what's important to you. It's not what's important to me. It's what's important to you. And so when you start talking about kind of clinical registries, again, who writes those? The physicians, the dietitians. And that's the information. And the way that that gets written is we see patients and we say, well, we see this really commonly, we should ask about this. We see this really commonly, we should ask about this. Now, remember, that's physician, dietitian, and companies. Okay, so then we're going to say, what's a regular clinical registry? So regular clinical registry is set up a priori, so prior to asking questions about things we already know and gathering information about things we already know. Okay, it does not say, okay, if it's not in the literature, but it's common enough, but it wasn't the most common thing seen, and patients all know this, so you guys talk on Facebook, you talk on all these things, and you're like, yeah, I have this, I have this. But if it's not published, it's not going to be in a clinical registry. It just isn't. Now, there's different types of clinical registries. There's the 10,000-foot clinical registry, and there's the I'm going to dive as deeply as I possibly can on this particular diagnosis. Okay, so there's those two types of clinical registries. Now, notice, though, who's doing the question and answering. What are the things I'm asking? When we start talking about clinical registries that are natural history studies that are originally written, often by companies, they are asking questions. What are the things that can change with therapy in this diagnosis? So that's and they make hypotheses that say, okay, we see, I'm going to say, osteoporosis in folks with homocystinuria. So I would think that if our drug impacts osteoporosis on a long time, I can use these biomarkers. So I'm going to follow these biomarkers. Now, those biomarkers may not be described in the literature for the rare disease, but they're following those biomarkers because they're looking for something that might move. Okay. All right. I suspect that's what you're going for, Danae. You want me to talk about EHOD too? You're muted.
Thank you. I did that so I wouldn't be noisy, even then I forgot. So I had a couple of questions I think I did in between there, but I think you actually hit all of them. I guess the only one I don't think that we hit on is, how, what is the difference between those things in an actual clinical trial? Because they're all different ways. So you have rare X, you have a clinical registry, we have a natural history study. Now we have clinical trial. So what is a clinical trial?
So
and how does that differ?
So clinical trial is actually a big umbrella. It can include natural history studies. It can include rare X. It can, because it's using patient information here. But what you're really asking is a clinical therapeutic trial, is my guess, right? So what's the difference between a clinical therapeutic trial? So usually when we say clinical trials, we're talking about, we have an idea that this particular compound can make this disease better, right? That's basically a clinical trial. And the question is, does this particular compound make the patient better? And so there's 3 phases of a clinical trial and kind of a fourth. The first phase is it safe? And it's usually tested on people without the rare disease. So that's phase 1. It can take a while to look at this. The second type is phase 2, and that's what's the dose? And a tiny bit, does it make a difference? And what biomarkers can I follow if I'm the company or if I'm the clinical trialist that change so that I can go to the FDA and say, look, this drug works? And phase 3 is really the trial that proves that that drug works at the dosing that was determined in phase 2. And so when you think about your eureka moment, I have this idea, to marketing this drug's on market, you're talking probably close to 25 years. I mean, I think it's important to think about that. Half of that is going to be the clinical trial time. And clearly, clinical trials only work as well as recruitment does.
Yes.
Right? So if you can't recruit patients to participate, and remember, we don't know, we think this drug works, but we don't know it works. And so that's kind of scary for everybody. But when you're talking about a rare disease, no matter which rare disease it is, it's rare, so there's few people. So looking at that and choosing the right biomarkers, which is usually informed by the natural history studies, the clinical registries, all of those things. And biomarker is just something that changes.
Yeah.
Okay. That shows how well this thing works.
Yeah.
And then I said there's a kind of a fourth. The fourth in rare disease is a phase 4, and this is a post-marketing study. And it's picking up more safety information because the clinical trial is probably really small because there's so few patients, that the phase 4 would be the post-marketing. And most rare diseases are on a pathway where there'll be a post-marketing study as well. So for example, drug is approved, somebody has a complication. It may be related to the drug, it may not be related to the drug. The patient may have gotten the flu, got hospitalized. If that drug is still in post-marketing, I get a fax with paperwork that I have to fill out to report back to the company. And so that's part of phase 4.
That is a
Danae,
very important part.
One
Yes.
thing that so clinical interventional trials, like Kim just discussed, they are often and usually in metabolic disorders, sort of medication driven.
They don't have to be.
They
don't have to be.
Yeah, they could be. So it could be any intervention that's being adjusted and specifically looked at in the study population. So like as a dietitian, it could be that I say, what happens if we change the protein ratio for people in this subset of the disorder? Like, what does that look like for these biomarkers that we've identified? And then those studies would still have the same issues kind of with recruitment, but they would be much shorter in duration typically, and you may be able to drive some sort of efficacy or non-efficacy of that intervention a bit quicker. And then so sort of as like an umbrella look, like a natural history or clinical registry is going to look at how the patient is already being treated wherever and however they're being treated, whereas a clinical interventional or therapeutic trial is going to look at how a new therapy affects kind of the same biomarkers or clinical endpoints.
And then there's a whole nother group of clinical trials, and these clinical trials have an intervention, and that may be like an MRI scan, a special MRI scan. Can I diagnose this? That might be an exercise regimen. Does this make people better? And people don't always think of those things as clinical trials, but those are clinical trials as well, because I'm doing something to the patient as opposed to a registry where I'm just reporting what is occurring.
Yeah. And maybe you can talk about this a little bit, just because, in homocystinuria, clinical trials are still new, and I know why we didn't discuss this in our questions, but that's okay. When you're typically doing a therapeutic trial, do you adjust your existing medications at all, or is it typically you continue your treatment, or is it differ?
So it depends. So it depends on what the protocol says.
Okay.
All right. In a lot of cases, you leave things the same. However, there are therapeutic trials in which you're supposed to take something away, or you're supposed to adjust things. So it all depends on the protocol. And that's a question the patients need to ask the person who's doing the trial is, okay, what am I supposed to do here? Am I supposed to change something? Am I supposed to not change something? It's a big conversation. So when a lot of decisions about how you design a clinical trial is a bunch of people locked in a room. It may be a virtual room, it may be a real room, but you get locked in a room. Okay. And there are discussions about, do we change this? Do we leave it the same? Are we going to have to add medication for this or that reason? What happens if somebody, I don't know, somebody goes off their diet? What happens if they have this particular complication? What happens if, and that's just a bunch of people brainstorming and coming up with how they're going to deal with those kinds of things. Something like an MRI scan, they may want you off your diet and on your diet to look at differences in the scan. But again, that's a protocol driven thing. So the protocol itself tells you what you're supposed to do. And those are questions that the person who's consenting you, the PI or the research coordinator, can review with you, and are questions you should ask before it starts, because sometimes those are, what is asked of you is something you're not comfortable with.
Yeah, the reason why I ask is I know there's this assumption, and it's a wish, you know, with the current clinical trials for classical homocystinuria, that, you know, patients, they're really excited about, you know, one day being able to leave the low protein diet and medical formula behind. And so there's this assumption that, you know, they get to join the clinical trial and poof, the diet goes away. And
for many of the medication trials, we tend to work very hard on keeping diet a controlled variable for most of the years of the study. And I think, sort of to Kim's point, there's a long period of time of work between the patient and the study team, kind of whoever that involves, that they come to know your baseline and your standard, and then we often are trying to keep certain aspects of care controls throughout the study, so kind of keeping those the same.
Because remember, the ultimate thing is, does this work?
Yeah.
Does this work? And I'm going to use an example. So does it work? So let's say we are using drug X, and drug X drops our homocysteine. Doesn't really matter what disease we have, because we're talking about homocysteine, but drug X drops your homocysteine, or at least that's the prerequisite, right? It's going to drop your homocysteine. And let's say when you drop your homocysteine, you feel better, right? And let's say you're on a prescribed diet, whether it's a low protein diet or just a prescribed diet a priori. Let's say if you change that diet and your homocysteine creeps up for whatever reason, then it's harder to show that drug X worked. And so that's why the decision, now there are trials in which one of the moving components is your diet changes. As you see whatever your marker is, we're going to say homocysteine goes down, you're allowed something that is not part of your prescribed diet, whether that's you're allowed more protein. And the change in the diet is what is the biomarker that they're actually following and maintaining the same homocysteine level. So like it's a decision prior to the design. And that decision isn't just like the researchers, that decision is also through FDA and whether they can see a statistical difference. But the ultimate goal of clinical trials is to show that something works.
Yeah, I think I appreciated that you also added that, you know, it's not just up to the company that's working on the drug, but it's also the FDA that helps make these guidelines and gearing them, because it is, the more I get involved, the more things open up to our patient community, the more I understand it. And like that, you just think it's just one side steering the ship, and it's really a whole bunch of people involved. So, and that includes the patients too, actually. Well,
patients can make a big difference with the FDA.
Yes, they definitely can. And so let's go back to the RareX registry here. And, you know, I know there's similar, not similar, but there's different groups that, you know, pull into research, and RareX, the Homocystinuria Data Collection Program, could impact their work. So Kim, could you tell us a little bit about EHOD, and then tell us about the work you do with them, and then how patient plays a role into that?
So EHOD is the European Network and Registry for Homocystinuria and Related Disorders. It was originally founded probably 10 to 15 years ago via the EU. So the EU put out a call for databases and guidelines to be written for rare disease. And EHOD is actually one of the grantees or grant recipients from the EU, which is why it's called the European Network and Registry for Homocystinuria Disorders. It is not just European. And I want to highlight that it's not just European. It's actually an international group, but because it was originally funded by the EU, it started as an EU. So the network is a group of expert institutions that work together to come together in terms of a database, and this database collects information that was determined 10 or 15 years ago about patients with homocystinuria and related disorders. So this includes the cobalamines, this includes severe MTHFR, this actually includes the folate deficiencies, and this includes MAT13. So it includes a lot of stuff that that aren't included in some of the other databases. And the idea is to collect information about what patients are doing right now across the world so that it becomes a baseline of information. So the last best natural history study prior to the recent EHOD publications was Harvey Mudd, who published in 1984 for cystathione beta synthase, or classical homocystinuria. That was the last good natural history study. So this is trying to bridge us, what does that end up being, 16?
It's
30,
38
35
years.
Yeah. So this is trying to, like, there's been a lot that happened in the last 38 years, including newborn screening and a bunch of other things. So it gathers all this information. It is limited, and it's from a physician perspective. So there's some labs in there, there's some outcomes. The questions are all related to the classical presentation of these disorders. There's a little bit of wiggle room for putting new information into it, but what is true and what's untrue in EHOD is difficult. It becomes the basis for this data collection, and a number of publications have come out of this over time describing the cohort. So if you look it up on PubMed, you can just look up EHOD and you're going to get a bunch of publications with it. And it's describing what folks have, basically. There's a little bit of intervention things in it, but not very much, because there isn't a lot of comparison between institutions, because every institution only has a few patients. Okay. Out of the requirement from the EU was guidelines, and these guidelines for these disorders. So there's a guideline for CBS deficiency, and there's a guideline for cobalamines and severe MTHFR. Out of that requirement comes a publication of guidelines for what's the appropriate therapy. Those guidelines have to be based on literature. They have to be based on publications. So there's some expert opinion in it. There's actually a lot of expert opinion, because in a lot of things that we do it because we were trained to do it that way. But it's like publications. So it comes out of publications. And those guidelines have to get updated every 5 to 7 years. So we're getting ready for the literature pull for the next set of CBS guidelines. And I know that the literature pull for cobalamine and severe MTHFR is ongoing right now. Okay. I'm not sure that's an answer, but let's talk about Rare X and what makes it different. So EHOD is doctors and physicians and genetic counselors and dietitians all get locked in a room, and we decided what questions we thought were important to take care of patients. Rare X is a series of questionnaires in which patients have figured out what they think is really important and gathers information. And it becomes another source of information. And I'm going to use an example because I was really lucky. Danae had me go through one of the original data, kind of the first data poll. And one of the things that became very apparent is dental disease. Like dental disease. I did the original CBS literature poll. There's a publication about dental disease. One. It is not any of the questions that are in the EHOD database. And I know this because I put stuff in the EHOD database, because the way that things go into that database is some poor physician or their staff types things in. And I mean types things in. Fills out a bunch of questionnaires, like click, 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 type, 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 click, 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 type, type, type. And so my time is what ends up in the database. So I have to pay attention to doing it. Rare X, when we pulled this literature, there's a bunch of dental disease. And so Danae and I talked to the folks in Rare X, we're going to pull this data at the end of November. So anyone that's registered for Rare X, please, please do your dental questions.
It's
Please, please,
called
please.
the Oral Health Survey.
And that's because we need to have it published before I pull the next EHOD data, because we need to include it, because it's something we're not asking about. Or if we do, we just kind of put it in the review of systems and it doesn't end up in the greater literature. We knew about the orthodontics, but just like dental stuff.
Yeah.
Okay. So that's the difference between Rare X. Rare X has like patient stuff in it, and it has patient questionnaires. It has what you fill out in terms of the questionnaires is what's important to you. Although I just asked you to do the oral health one because I think that that's important for us all to know.
I think it's just a little bit more of a holistic approach, like
Yeah.
you know, because even the things that aren't documented are asked about. And I think that sometimes what gets a little bit missed is that, you know, we only ask questions about the things that we have documented as known problems. And unfortunately, more things pop up. And I think part of it too is patients are living longer, and, you know, our problems are starting to look a little bit different because of that. And also, you know, you think 84 publication treatment affected patients very differently then.
Yeah,
well.
I mean, like, I don't think people realize the natural history study that almost all the guidelines that were published, I don't know, 5-6 years ago, is the 84 publication. That was where a lot of that data, a lot of the underlying data came from.
Yeah. So I'd like to transition a little bit here, because we've talked about the clinical standpoint and, you know, how this affects geneticists and how they treat us, and, you know, how that shapes their research. But Suzanne, can you tell us a little bit about GMDI, the work you do with them, and, you know, any of the other, because I know that you're a research dietitian, which there's really not that many as far as I can tell. And so can you tell us a little bit about that? And, you know, I think CERN also writes the guidelines for nutrition. So I don't know if that's, there's this umbrella of things that I'm trying to still wrap my head around. So I don't know if it's CERN or GMDI, but maybe you can talk about how the nutritional guidelines get written.
Come to be.
Yes.
How they come to be.
And how patient data plays a role in that.
It's a great question. So I think GMDI, let me just like take a step back. GMDI is Genetic Metabolic Dietitians International, and it's an organization of metabolic dietitians with the goals of kind of enhancing research, enhancing clinical practice for metabolic disorders. And supporting metabolic dietitians, because like you said, I think in general, we're a rare breed. And then those of us who participate in research are even fewer. But the most significant connection that GMDI, I think, has to this conversation is that there are published nutrition management guidelines for certain metabolic disorders. And this was a project that was started a number of years ago, and the goal was to go through sort of each metabolic disorder that we often see and create a robust set of nutrition guidelines so that our practice standards are standardized. And so once we know where everyone is practicing, we can better improve what the practice actually is going forward. We can study it kind of more consistently. We can figure out like, oh, in what situations does this not apply, and sort of try to capture the heterogeneity of different patient subsets. So the way the guidelines come to be is that, much like Kim just described, it's a literature, it's kind of a scouring of the literature to start, and that really serves as the basis. I think for the nutrition management guidelines, they really recognized in putting together the system that they create that there is a large amount of unpublished literature, or what we're calling gray literature, which is sort of communication back and forth amongst professionals often, or presentations and things that we've seen. So they pull all of that together. And then after they pull all of that together, they go through a few different series of polling expert opinions. And again, though, those experts that they're polling are mostly healthcare providers. There is one expert opinion sort of coming together of expert opinions that does include patients or families at one point in it. I think we all recognize that nutrition interventions only work if people can do them at home. And so that was an important component of one of the expert polls, but it is largely, like Kim explained, for the other guidelines, provider driven in scope. And then at the end of that, guidelines are published. But I think the goal is that the guideline is meant to be, a living document and something that we're kind of constantly coming back to as like a basis of care, but moving, kind of evolving with both patient individuality and then also really trying to study, when does this not apply, or is this guideline really no longer valid as things have moved along?
Can I mention, I think this is a good example. So I know in the PKU community, there's been different nutrition guideline sets throughout time because of different drug introductions. And so we all think that once the standard is set, it's set. But the truth is that as research and development and therapies evolved, our standards of care and the guidelines for nutritional management are also going to change too. So all the more reason for,
the more that we know, the more that we understand, the more that we have therapeutics or interventions or different diets, this has got to be a living document. It's got to be revised because there's new information that comes up all the time.
Mm-hmm.
And so I'd like to highlight that it's got to be a living document. There's no way for us to take care of you if we don't improve what we're doing.
Yeah. And I think that's one of the things that is important that people understand, like with RareX, with the data collection program for homocystinurias, is it's not a one and done. You need to go back each year and update your surveys. If you go have your gallbladder out, go in there and
Please.
Update that. If you start a new medication, go into the medication survey and add that. Take out a medication that you're no longer on. There's, we don't just stop at our baseline because our bodies change over time. So
can I use some examples of things we don't know? Because I don't think people know these things that we don't know. So let's talk about total homocysteine. Okay, let's talk about total homocysteine, and you can apply whatever disorder or whatever rare disease you have, okay? We don't know whether we do know that it varies around a mean. We don't actually know for sure if that mean changes. And what I mean by that is currently we have flashes of individuals at different ages about where their homocysteine is. And remember, if you have had homocystinuria for 50 years, your therapy is different than someone who's had homocystinuria for 5. And what we've done interventionally, with the diets, with the recommendations, with the therapies that were available to you is different. So I can say, okay, my 50 to 60-year-old classic homocystinurias typically have this homocysteine. I don't know if that would be the same homocysteine for a 5-year-old or for that when that 5-year-old is 50. Because what we don't know is what's the normal homocysteine for somebody with a homocystinuria rare disease.
Yeah.
At what age?
Yeah.
Yeah. And
or what's the ballpark? Where should we aim?
Yeah. And sort of similarly thinking about the evolution of the population as a whole, and this doesn't even get into that every individual is different and their presentation is different. We've only been newborn, we haven't had newborn screen around kind of for all of this time that we've been collecting data. Yeah. So things like infant feeding, I think, are really relatively new to the field. We don't know what it looks like to kind of employ different modalities of infant feeding or exactly what that kind of looks like treatment wise. what it
And
looks
what
like
and what it's
acutely
going to
and
look like in
chronically.
10 years.
Yeah.
Like, what is it going to look like for kids that are infants now that we're feeding this way, and what's it going to look like for in 10 years? Because we don't have any of that information. And this is basically a long way to say, please update your information, because we don't, because that linear progression, what we really have is snapshots in different generations of folks.
And I think, like, Kim, I'm sure you'll agree, like, we as providers understand that the treatment's only effective insofar as it's utilized by patients. So what we can prescribe things and ask people to do specific things, but we're always kind of benchmarking off of what we prescribe. But if we understand, for example, that this diet intervention or this medication is impossible to give the way it's being prescribed, then we can start to sort of change recommendations based on what the actual patient experiences.
I think that's
And
a really good, I just wanted to say, I think you guys make such a good point, because I've always encouraged, like, as patients, sometimes we feel like, you know, we don't want to be nagged because we know we're not doing a great job. So it's, so a lot of times you just, you know, you cheat your diet log, you aren't honest. And then, but the truth is, it only hurts you as a patient. And I think that's like one of the biggest obstacles in, you know, trying to improve things. Because if you're not being honest with your dietitian about what you're eating, they can't help you navigate ways to make it easier. And same with the geneticist, like they really don't know what's going on when, you know, you aren't following that prescribed plan.
I can only believe what you tell me.
Yeah.
I can be suspicious that you're not telling me the right thing, but I can only believe what you're telling me. So, like, if I'm increasing your betaine over and over and over because your homocysteine's drifting up, but in reality, like, you're cheating on your diet, I don't know that.
Yeah. Or if they're even taking their betaine to begin with.
That's exactly right. Or if because it's gritty and slimy, they're not.
So you know, and that's the thing. It's just, I think you need to be honest with your clinic, and you also like tell them the things that you're going through. I feel honestly like my genetics clinic listens to me better than my regular primary care doctor. And I think it's because they're used to seeing a more diverse set of patients with more demanding needs. So your clinic is really there to your benefit as a patient too. It's not like, not just for the homocystinuria side of things, but you as a whole patient. I know they've helped me navigate so many more issues. And then it's like, oh, then they're like, well, if your level is a little bit higher, this makes more sense now. Like when you start actually sharing more with them. So it's not just about going in and, you know, sharing that diet log and all these other things. It's there.
Did you have a cold? Were you newly diagnosed with, you know, inflammatory joint disease? Did they add a new medicine?
Yeah. All those things are so important. I think you were going to say something, Susan or Kim, and I interrupted, but I just was like, the honesty part is it's such a major thing for me, because I don't think patients realize that it has such an effect in their care. Like your care is not your care.
And it affects research, because if we know that the treatment doesn't work for a large subset of people because it's hard to do, that means that we need to continue to improve the treatment. And like these types of surveys that give us insight into what's actually happening on the patient side are critical for us to kind of help formulate treatments that are going to be more approachable, improve quality of life. Like that's kind of ultimately always the goal. It's even something sort of as kind of rote or mundane as like formulas changing. Like if everyone just tells us they're taking the formula, but then we aren't going to be motivated to change what that looks like or how accessible it is, or, you know, so we want to always increase people's ability to do the recommendations that we think are working well, and then we'll continue to study those recommendations as well.
Yeah. I was looking here at my questions here. And so we talked about how, it can affect our therapies and our outcomes as actual patients, but what we haven't talked about is how it affects patients in terms of newborn screening. So I have a question here. Would the standards of care look different if we knew how many patients were being missed by newborn screening? My question doesn't make sense, but I know what I meant. It says being missed by being diagnosed. So either delayed or through newborn screening. And, how does that affect what you guys do? And how would how can RareX affect that?
So I think it's important to know that we don't know. So we as your providers know whether you've been missed by newborn screening or not missed because we probably did the diagnosis. Okay. I don't think people realize in general, like general physicians, general medical professionals in general, they don't realize how many CBSs and how many MTHFRs and cobalamin Cs are not identified by newborn screen. Okay, so one of the issues is being able to document how many people are identified by newborn screen and how many people are not identified by newborn screen. So RareX actually helps us do that. So the other thing is, let's talk about severe MTHFR, because I'm going to use that. So it's a low methionine. Most states don't look at a low methionine, but there's a state to state to state to state difference. So everybody's states does a newborn screen slightly different with different things, but in general, everybody does it about the same. Their cutoffs are all different. So if you were born in state A and somebody else was born in state B, and state B is looking for low methionines and homocysteine, if you were born in state B, you might have been picked up by newborn screen, and you may not have been picked up by newborn screen by A. I know that as a geneticist and a metabolist, and we have 4 states we do newborn screen, so I know which states do which parts. But in general, we can't go back to the states and say, look, this is the number of people that are not identified with this diagnosis by your newborn screening. These are the folks that were missed with your cutoffs. And so that helps us figure out which newborn screens are being missed, because there is not a database that says this is the people that have a metabolic disorder that's on the newborn screen. These are the people that have been screened, and these are the people that have been missed. There isn't any like national, international thing that's that way. Moving on, so let's say you were picked up by newborn screen, and we initiated therapy in the first 2 weeks of life to month, 2 weeks to month. What does your outcomes look like versus that person who was not picked up by newborn screen? And was that person not picked up by newborn screen because it's not newborn screened, or were they not picked up by newborn screen because the cutoffs were different? Right? So we have no way to be able to do that kind of tracking in our current mechanism of data collection. I suspect that's where you're going with this.
Yeah.
And so HCU Network America is aware of the number of people that have reported to them that they've been missed by newborn screen. But the states in which they were screened are not always aware of that, because I, as the provider, may not have reported back to that state, or may have reported back to that state when I made your diagnosis.
Yeah. I think that's the really important thing is, like, if you are a patient or a caregiver and your state had newborn screening when you were born, all states had newborn screening as of 2009 for classical homocystinuria.
But it differs, like different states have different timings of when they started newborn screen.
Yeah.
So you may know that you've been screened, but you may not know if you've been screened for classic homocystinuria, or maybe for MTHFR and maybe for cobalamin C.
But it can but you can have your genetics clinic. impress upon them to report back the missed case to the lab, and they need to know that for their research and development too, so they can adjust their systems to help flag patients. I know that goes beyond rare X, but you know, this is why it's important for us to have that data and for them, so we can go
but
to
I think,
them and have those
Danae, I think
conversations.
this is like a really good point in which like patient-driven data and sort of capturing this can help us smooth some of these gaps that we have in kind of a state by state system, and some of the data collection problems that we as healthcare providers have not yet been able to solve. So it can push advocacy, it can really drive change to have this data captured well.
And if you put your data into rare X and you were missed, we can actually do that as a composite piece of information, and we can go to the national meetings and we can say, look, we've got this natural history study that's going through rare X, and look, this is the number of people that were newborn screened that were not identified on the screen with this disorder, this disorder, this disorder. And then we can file in a little bit closer and say, this is what makes these people more related, or this is what makes them different from the folks that were identified at the same time. And then we can ask the question, how does newborn screening improve outcomes? Because our aim is newborns' early diagnosis and management improves outcomes. And so like, then we can say, look, these folks who were screened identified had these outcomes versus these folks who were later identified. And we grant that these folks had this and this that are different, but like see where the differences are.
Yeah. I think that's the really exciting thing is like, you know, Kim talks to patients, I talk to patients, Susan talks to patients, and we know of these cases like where they were caught by newborn screening, especially for like some of the cobalamin disorders and in severe MTHFR, and the outcomes are so different because of newborn screening. So even in the case that you were flagged, it's super important because right now, a lot of those disorders are not on like the newborn screening panels. And
they're not on every state.
Right. But if we have that data say, look, these patients are doing well because they were diagnosed, that's an argument to the state, to the federal grouping that it's called the ACHDNC. It's a long acronym, but but it's a good argument to them too. So we can point out, you know what, newborn screening does work in these cases. We have outcomes
data that demonstrate that these patients do better than these patients.
Yeah. So, you know,
even though there's going to be individual differences,
missed and not missed, you're all important in this, in helping newborn screening move forward. And so I know we only have a few minutes here and I want to be respectful of your time. I don't see any questions yet. But if anyone has questions, feel free to drop them into the chat or the Q&A. There were some comments online, but they're just excited. One of them says, the exciting message that I'm hearing is that we as patients and caregivers have the ability to drive the future of our treatment. And it's very much true. It pays to be an involved participant in your healthcare. And there's a lot of ways to participate.
And people have different comforts with research.
Yeah.
Right. So like, it's okay if you're like, I'm only going to do the natural history. It's okay where I'm only going to do rare X. It's okay if you say, I really want to do a therapeutic trial.
Yeah.
Right. So like, and then it's okay to say, you know, I'm really so not comfortable with research at all for whatever reason. That's okay too. But I want to like encourage people to participate wherever and however they feel comfortable.
Yeah. And you know, the thing is, it's not a one size fits all. There are going to be patients. Number one, there has to be if for clinical trials, there has to be one to exist for you to participate. And then just because it exists doesn't mean you're going to qualify. The great thing I will say about RareX is anyone with any type of homocystinuria diagnosis, and, you know, no matter where you're at, you know, if your level's 500, which I really hope it isn't for your safety sake, or if you're in a normal range like myself, you know, there's a place for you here. And so that's the great thing. And natural history studies, you know, typically are condition specific. So, you know, there has to be one for your condition. So I feel like RareX just kind of lends itself to everyone pretty easily, and you get to do it on your time. That's the other thing I really appreciate, because I don't have a ton of time between work and family.
And I think regardless of your like comfort level with research, if you're interested to know what's going on, even if you yourself don't want to get involved, that's an important conversation to have with your providers in clinic. We, I think, often try to bring it up when we can, or when we remember, or when there's sort of large movement, but we don't always have that as sort of a standard part of our visit. So if you're interested and you want to know, ask questions, you're always allowed to ask as many questions as you want, and you don't have to participate in the end. You get to consent at every step.
Yeah. You get to choose. And so like asking questions as somebody that's interested in a study, or asking questions, what is the field bringing? What are we seeing coming forward? How close is this to approval? Sometimes we'll have a pretty good idea of that, you know.
Yeah. So I have a couple of questions here for you guys, not from anyone, but just questions. So number one, is there anything that you feel that we didn't touch on that patients should know when it comes to research, you know, how it affects what you guys do, how it affects the field, and then what are your hopes for the future for homocystinuria as a whole, or, you know, the rare disease community?
Yeah, I think one big thing, specifically as a dietitian involved in research, is that diet is notoriously difficult to study, not only in rare disease, but in the population as a whole, because most of us don't pay a lot of attention to like every bite that goes in our mouth. So I always encourage people, like as much as you can, both clinically, but also like anytime you're filling out surveys or doing research, to be honest, like we're trying to improve the diet that's actually being consumed, and not just the diet that is being prescribed. So I think with food specifically, a lot of us tend to write or say what we think people want to hear, or what we ourselves wish we were already doing and are not doing. But what we really need to know is where everyone actually is currently in order to make improvements.
And then I think I want to kind of focus on data is what drives guidelines, and data also drives companies to be interested in providing therapeutics. So rare diseases that have natural history studies or something like that provides companies a basis from which they can expand or can approach that particular disease. And the more involved patients are in that rare disease's research stuff, the more likely a company will get into rare disease. And I, as a provider, want as much in my toolbox to take care of you as I possibly can be. So I'm really excited that CVS has multiple players, that we're starting to see some in MTHFR. We're seeing more research in cobalamin C. And I think part of that is like there are guidelines, there's some natural history studies, but part of it is patient groups are being more and more active, and thus companies are noticing and want to get into this space. And so like, even if you don't participate in the clinical trials, being active in family groups like HCU Network America actually helps everybody But I, as a provider, and I'm sure Suzanne would say the same thing, the more I'm in my toolbox, the more options I have. And I'm totally fine that there's multiple trials going on within a particular disease. It makes it tough with recruitment enough patients, but like that gives me more tools.
Yeah.
And so I think that's what's really exciting is there's like people in the field. So that's pretty cool.
Yeah. I tell people like it's crazy to think where things are headed. And there's definitely
what was here 10 years ago versus 5 years ago versus now.
Yeah. And I think, you know, in another 5 years, it's going to be a totally different landscape than what we're seeing now, which is incredibly exciting. I know the only therapy that's been approved in my lifetime was cysteine, and you know, there's
6 patients. It was approved based on a publication of 6.
So we can do this with our community with all these new drug trials. So it's really exciting to see where the future is going.
And I think like our goal as healthcare providers generally is to provide individualized healthcare. And that's the same for our patients with inborn errors of metabolism. And so we put out these guidelines, and they're like, they are meant to cover a wide range of people, but they are not going to capture everyone. And the best way for us to capture the heterogeneity of any group is more participation. And so I think if you're someone who feels like you have not been well served by current treatments, your participation is really important, because we are not capturing you well or serving you well yet.
Yeah, I think that's a really important thing too, is just like, if you have questions and you don't feel like any doctor's been able to give you an answer, this is like a great place to like put the things that you're experiencing and hope that, you know, because someone out there has the answers. And I think if we group our experiences together, we're much more likely to find them.
Sometimes we have the answers.
Yeah.
Like I may look at you and say, I don't know the answer to that. I'll see if there's anything in the literature, because that's what like,
really
and I'll
good at
reach
that.
out and I'll reach out to my colleagues who like have been doing this for 50 years instead of 30. So like, but like if somebody says, I don't know the answer, but I'll see if there's anything about it, we're probably being pretty honest with you.
Yeah. And that's, I think, why we need more patients. We just really need more, and especially as the patient population ages, because it's going to look different. It's really exciting seeing some of these other updated guidelines for patients. I know we're at time here, so I could keep going. I appreciate the both of you coming on early this morning. Well, it's not as early for you, but it was early for me. So, but I thank you, Kim and Susan, for coming on and talking to us about the importance and how patient data can shape our standards of care. As you can see, your experiences you share with the RareX HCU data collection program will help contribute to a better understanding of homocystinuria, which leads to better clinical care. If you have not joined the RareX data collection program for homocystinuria, please head over to homocystinuria.rare-x.org and enroll today and start filling out those surveys, especially the oral health survey. So Kim can write her paper for us, because I know it's an issue, so I know other patients know
and the thing is, like, as I said, there's a publication, which incidentally is in Japanese. There is a publication. And so, like, it would be really nice to be able to get it into the next set of guidelines, at least highlight it.
Yes, well, that is so exciting to know our data is already being put to use. Well, thank you so much, Kim and Suzanne. And if anyone has questions, forward them to me and I can direct them to Kim and Suzanne when they are available. All right. Thank you so much.
Thank you, guys.